Hello friends, Janvi here. Welcome to my YouTube channel. Today I have come up with the third video on genetics. If you have not seen the part 1 and part 2 of the topic, you may refer to the description for the link and go through the same before proceeding to today's video. Before we start today's video, if you like my channel and tutorials, please subscribe to it and share it with others. Hello friends, today we are going with the next part of genetics where we will study about the structure of the DNA, structure of the RNA and gene interaction, the remaining parts of the gene interactions like multiple allelism and polygenic inheritance. Let's start about the structure of the DNA. DNA is called as a deoxyribonucleic acid. And we all know it's a genetic material which is present in all living organisms. And DNA is responsible for the passing of the information from one generation to another. There are three forms of DNA, mainly zDNA, bDNA and aDNA. The structure of the DNA is studied through X-ray diffraction, and mostly I'm concentrating only about the bDNA which is present in the human beings. When we talk about the bDNA is having a right-handed helical structure. It has 10 nucleotides per turn and anti-parallel strands which are present. That means the anti-parallel means one strand will run from 5' to 3' end, whereas another strand will run from 3' to 5' end. They have complementary base pairing. That means two strands have complementary base pairing with each other. There are two types of grooves which are present in the DNA, major groove and the minor groove. Let's learn about the complementary base pairing, anti-parallel strand, and also study about the structure of the DNA. So let's start with the complementary base pairing. What is complementary base pairing? See, in the DNA, the two strands of DNA will be present. If in one strand the base is adenine, in the adjacent strand the base will be thymine. And these two are bonded with each other with a double bond. The next example is, if there is a guanine in the one strand, the adjacent strand will be of cytosine, and these are bonded with each other with a triple bond. So the adenine will always base pair with thymine and guanine will always base pair with cytosine. One with double bond and another one with triple bond. This is called as complementary base pairing. Second one is anti-parallel strand. What is this anti-parallel strand? The two strands of DNA are present. I have already said about this. The one strand of DNA will run from 5' to 3' end. The other strand will run from 3' to 5'. One from 5' to 3'. Another one is from 3' to 5'. Hence it is called as a anti-parallel strand, and this is very important during the process of replication of DNA. Next thing is about the structure of the DNA. Let's talk about structure of the DNA. I have shown in the diagram, you can see one strand the blue color strand is running from 5' to 3' end, whereas red color strand is running from 3' to 5' end. There are two types of grooves which are present, major groove and the minor groove, and the diameter will be around 10 angstrom. And you can see I have shown certain nucleotides which are arranged that are in yellow color. So the 10 nucleotides are present in particular turn. So per turn you can see about 10 nucleotides which are arranged in the DNA structure. Structure of a DNA in detail with the basic concepts. When we talk about the DNA, we all know that DNA is made up of deoxyribonucleotide. So when we talk about the ribonucleotide, deoxyribonucleotide, what are the things which are present in this deoxyribonucleotide? The deoxyribonucleotide is made up of three molecules, namely a phosphate group, a pentose sugar, that is nothing but a deoxyribose sugar, and also a nitrogenous base. Any base which is attached to the nitrogen is called as a nitrogenous base. And there are four kinds of nitrogenous bases, namely adenine, guanine, cytosine, and thymine. Together it will constitute purines and pyrimidines. Always remember, purine will bond with pyrimidine. So adenine will always bond with thymine and guanine will always bond with cytosine. Structure of the phosphate and the sugar. We can see that for phosphate is bonded with the oxygen, where here they have to satisfy both the valency, that is phosphate valency as well as the oxygen valency to form the bond. And the second one is a pentose sugar, which is nothing but it can be a ribose sugar or deoxyribose sugar, but the differences will come to know in the next slide. But remember, pentose sugar is with the 5 carbon atoms in them, and pentose sugar may be a deoxyribose sugar or a ribose sugar. Let's see about the structure of the nitrogenous bases. Here I am showing the structure of cytosine, thymine, adenine, and guanine. See, whenever we talk about the bases, namely whatever it may be, in common, in generally in human beings, the chemicals which are formed or the elements which are present are normally a carbon, hydrogen, and oxygen. In addition, you may have like nitrogen also. So in certain bases or the bases in which nitrogen are attached are called as nitrogenous bases. And you all know the basics of chemistry. Any element to form a bond with another one, it has to form its valency. It has to complete its valency. Based on the valency and other things, the mainly carbon has to satisfy its valency, nitrogen has to satisfy its valency. Depending upon all these things, the bond is also formed in each bases. Mainly it may be a cytosine, thymine, adenine, and guanine. So you can see in between double bonds, single bonds, double bonds, single bonds which are present along with the carbon hydrogen or carbon nitrogen or carbon oxygen, depending upon the valency and how they satisfy their valencies. So in general, in this slide, we will talk only about the structure of nitrogenous bases. As we all know, nitrogenous bases are made up of carbon, nitrogen, and hydrogen with a double bond or with a single bond, or alternative single and double bond to satisfy their valencies and to exhibit a stable configuration. The nucleotides are made up of sugar, phosphate, and the nitrogen base. Now we will see how the structure of the nucleotide will appear. I have shown here in the diagram 1, 2, 3, 4. 4 nucleotides are arranged one below the other, wherein they are connected with one another with the help of the O. The bonding is formed with the help of the O. So let me explain you in detail. See here, the first nucleotide is having the deoxyribose sugar, and the second nucleotide is also having the deoxyribose sugar. The link is in such a way that the carbon 3 of the first nucleotide sugar will be linked with an carbon 5 of the second nucleotide sugar. The same way, the second and the third nucleotide are linked with one another. The carbon 3 of the second nucleotide sugar will be linked with the carbon 5 of the third nucleotide sugar. This is the way through which the bond is formed between the n number of the nucleotides. Hence they form the structure of the nucleotide, and this is the structure which is seen in the DNA molecule. That means this is how the structure will look in the DNA molecule. The bonds present in the DNA. There are two types of bonds which are present in the DNA, which we'll call it as a glycosidic bond and a phosphodiester bond. What is a glycosidic bond? The bond which is present between the deoxyribose sugar and a nitrogenous base is called a glycosidic bond. The bond which is present between the phosphate and the sugar is called as a phosphodiester bond. And the two things which you have to remember to understand the concept of the structure of the DNA, mainly the nucleoside and the nucleotide. What is a nucleoside? Base plus deoxyribose. That means, for an example, I will take adenine as a base and sugar, a pentose sugar, which is deoxyribose sugar, together form a nucleoside. Whereas nucleotide is nothing but base plus deoxyribose plus phosphate group, which is attached to the base and the sugar. Hence, we will call it as a nucleotide. This is for, actually they may ask in certain papers also. So be clear with this nucleoside and the nucleotide. Structure of the RNA. The another important genetic material which is present in the most of the viruses and also in some plants. The RNA may exist in two forms. Either it may be a single stranded or either it may be a double stranded, but it's not a helical structure. The virus which affect the plants are called as plant viruses. Example, the tobacco mosaic virus, which affects only the plants and have single stranded RNA in them. The influenza virus is the virus which affect the animals and it is a double stranded one. So when we talk about the RNA may be single stranded or it may be a double strand. Next thing is the virus which transmit or which acts RNA as a genetic material is called as a genetic RNA. That means RNA can exist in two forms. In some forms, it acts as a genetic material which carries the information from one generation to another. Hence, we will call it as a genetic RNA. But for example, if we consider human beings, we all know that most of them know that RNA exists in three forms. It may be a messenger RNA, it may be a rRNA, or it may be a tRNA. And all this will help during the process of transcription and translation. We will explain transcription and translation in the later process. But at present, remember, in all these types, the RNA doesn't have any role to play with the genetic material. In all the cases, it is not involved in passing the information, but it agrees the information which is given by the DNA. Or we can say it obeys the information which is passed by the DNA. So here, whatever the information the DNA will provide, according to that the RNA will work. Hence, we will call it as a non-genetic RNA. So remember, the RNA is of two types. Once again, one is genetic RNA and another one is non-genetic RNA. The genetic RNA is the one which transmit the genetic material from one generation to another, whereas non-genetic RNA just obeys the order which is given by the DNA. And three forms for RNA as non-genetic RNA are mRNA, tRNA and rRNA. RNA is very similar to that of the DNA. The only difference which exists between the RNA and the DNA is with the sugar and the nitrogenous bases. There is few difference which we can see when we come across the RNA and the DNA. For example, in DNA, the deoxyribose is present, whereas in RNA, the ribose sugar is present. The RNA does not have a helical structure. Apart from that, when we talk about the nitrogenous bases, thymine is present in DNA, whereas uracil is present in the RNA. That means when DNA comes, the adenine will always bond with thymine. But instead of thymine, here it is replaced by uracil. So in RNA we will see uracil, whereas in DNA we will see thymine. So always remember, during the process of transcription, when the mRNA is getting produced by the help of the DNA, when adenine will come, the RNA, the messenger RNA which is formed, will code for uracil instead of thymine. Always remember this point. A will always bond with U in an RNA when it comes to an ribonucleic acid, which is nothing but an RNA with again a double bonded structure. Difference between the deoxyribose and the ribose sugar, which is very important to understand the structure of the DNA and the RNA. Why we will call it as a deoxyribose sugar and why we will call it as a ribose sugar. You can see in the diagram why we will mention as a pentose sugar, because it is having a 5 carbon atom. At the second carbon atom, we can see only at the down of the second carbon, only H is present in the deoxyribose sugar. That means instead of OH, the O is removed and only H is present in the deoxyribose sugar. Deoxy. D means removal of the oxygen from the carbon 2. Hence we will call it as a deoxyribose sugar. But when you see the ribose sugar, that the carbon 2, you will have H and in down you have OH. This is the difference between the deoxyribose sugar and the ribose sugar. Deoxyribose, the O which should be present will be removed, hence it is called as deoxyribose sugar, which is present in the DNA, whereas ribose sugar, where you have OH in the second carbon atom, is called as ribose sugar, which is present in the RNA. With this, we will end the structure of the DNA and the RNA, where you have studied in detail about the RNA in which the nitrogenous bases are present, what type of sugar are present, and whether the RNA is single stranded or double stranded, and you have also studied about the structure of the DNA. Together, by this we will end with the structure of DNA and RNA, and next I am going to continue with that is nothing but the gene interaction, where we have left with another 2 concepts which says about multiple allelism and also about the polygenic inheritance. We will be continuing the gene interaction session here also, in which I am taking the 2 examples to complete the gene interaction. They are multiple allelism and also polygenic inheritance. Now we will start with the another 2 types of gene interaction, which we will call it as a multiple allelism and another one is polygenic inheritance. Let's talk about multiple allelism. When we talk about the multiple allelism, multiple means what? More than 2 genes or many genes. Multiple means many. Here, more than 2 genes will interact to produce a particular character. Multiple means many. The same thing applies here. Multiple allelism means multiple allele, multiple genes will interact with one another to produce a particular character. So, the best examples to explain the multiple allelism is a blood group which is seen in human beings. We all know that the blood group of an individual will be either A, or it may be B, or it may be AB, or it may be O. Anything, any of one of this will be present in the blood group of the human being. But how the blood group is produced in an individual, how it is characterized. Remember, the blood group is decided with the help of the gene called as gene I. Remember, the gene I is the one which will decide a particular blood group in an individual. And 3 alleles are responsible for the blood group. They are A, B, and O. These three are very, very important. Three alleles, namely IE, IB and IO. These three, mainly A, B and O, are important to decide the blood group in an individual, and the gene which is responsible will be an I. So let us explain about the multiple allelism with an example of a blood group in detail in the next slide. Let me explain the multiple allelism in detail. First, we will start with an RBC which is present in the blood, which we will call it as a red blood corpuscles. So, when we talk about the RBCs have antigen in them, or you can see in the diagrammatic representation I have shown an antigen A with the red color indicating the A. A means what? The antigen which is present on the RBC. So when we talk about an multiple allelism, remember the RBCs have a particular antigen on them. The antigen which are present may be antigen A or antigen B, or in some RBCs both AB antigen are present, but in some RBCs nothing is present, and you can see clearly what type of antigen is present in what RBCs and what name are given. If antigen A is present, it is called as A blood group. If antigen B is present, then we will call it as a B blood group. If both the antigen A and B are present, then we will call it as an AB, whereas nothing is present, then we will call it as an O blood group. See, you can easily distinguish this using a diagram which I have indicated here. But when we talk about a multiple allelism, always remember the blood group will come either from the maternal or the paternal features. That means either the blood group should come from the father or it should come from the mother. To analyze this further, we have to do certain crosses between the parents, that means the father and the mother, to study about the blood group in the offspring. Let's see in the future slides about the cross which is done between the parents and what type of blood group is seen in the individual or the offspring. Let me explain about the multiple allelism by taking an example. Here I am taking a parent, the father and the mother. The blood group of the father is B and the blood group of the mother is A, which I have indicated in the brackets. The allele of the father will be IB, IO, whereas the allele of the mother will be IA, IO. The gametes which are produced will be B, O and A, O. When they are crossed with each other, the offspring, what the blood group will be received by the offspring is, when we do the cross which I have indicated in the diagram or the representation, you can see the cross which is done between the B and A and produce AB, B and O produce BO, O and A produce AO, and O and O will produce the O. That means AB blood group may be present, B blood group may be present, A blood group may be present, or O blood group may be present in the offspring. Either AB or B or A or O, anything can be present in an individual. Multiple allelism with another example. Here we can see father with a heterozygous B blood group and mother with a homozygous O blood group. When both are crossed, the gametes which are produced are IB and IO, and for a mother IO. So the offsprings which are produced will be heterozygous IB, IO and the homozygous IO and IO. So the blood group of an offsprings will be either B blood group or an O blood group. The another one example to explain the multiple allelism. Here the father blood group is homozygous B and mother's blood group will be homozygous A. The gametes which are produced will be IB and IE. When these both are crossed, we will get an offspring of IAB, which we'll call it as a blood group AB. That means the offspring is having a blood group with an AB in them. You can see here on this slide I have given few problems for you people to solve. Please go through with it, where I have given you the 3 problems for you to understand the clear concept of multiple allelism, where you can see first problem is with parent with IA, IB and IB, IO. The second one will a parent with IB, IB and IB, IO. And the third problem is parent with IA, IB and IB and IO. Please solve all these problems and crosses so that you can understand the clear concept of multiple allelism. Let's talk about the polygenic inheritance. So what is a polygenic inheritance? The word itself says again the many. Many means poly. So many genes will interact with one another to produce a polygenic inheritance. It is a quantitative type of inheritance. Remember, it is a quantitative type of inheritance. That means it depends upon the number of the dominant genes. That means the dominant genes will decide the character of an individual, and all these are present at the same loci. So the 3 genes which control the skin color in the human beings are A, B, C. So the polygenic inheritance, the inheritance which is seen where the interaction is between more than one gene. That is, many genes will interact, and mainly the dominant genes will interact. The dominant gene, the number of dominant gene will decide the which character to be produced in an individual. To study this, the best example which is quoted is the skin color in the human beings. We all know that the pigmentation is mainly due to the presence of the melanin which is present in the skin. So the 3 genes which code for the melanin pigment are A, B and the C. You can see in the diagrammatic representation where I have given you the dominant AA, BB, CC, and here there is an intermediate AA, BB, CC, whereas full recessive character AA, small bb and small cc. In dominant, all the letters A, B, C are dominant and they produce a dark individual, whereas in the intermediate you can see capital A, small a, capital B, small b, capital C, small c, and in recessive you can see full recessive character A, B, B, C, C. All these are in small letters in the recessive character. So the recessive or the small letters will indicate those are very whitish, whereas the dominant with all the 3 genes are dominating, then the individual will be the dark. So this also play a very important role in deciding the color of the skin. In few the skin color will be white, in few the skin color will be black. This is mainly due to the presence of the melanin pigment. So those who are having the melanin pigment at the maximum amount will be the darkest ones. The 3 genes, that is A, B, C, all the 3 if they are present in the dominating condition, then whatever will be produced, the individual will be having the maximum amount of melanin and they look very dark in color. Whereas if all the 3 genes are recessive in condition, all the 3 genes A, B, C are recessive in condition, then the individual will take up the white color with no melanin pigment. So if all the 3 genes are dominant, they produce a dark color with maximum amount of melanin pigment. Whereas if they are recessive, they are all having an small letters A, B, C and they are not dominating, hence they produce a white color and the melanin pigment is absent. But the intermediate which are produced will be either they will be of 50% maybe a dark or 50% white, depending upon what type of genes are dominating and whether the one gene is dominating or two genes are dominating or three genes are dominating. Either it is an homozygous condition or it's heterozygous condition. Based upon all these things, they will decide what type of color may be taken up by an individual. So this is a quantitative type of inheritance where we can see that how many dominating genes will decide the skin color. The maximum 3 will be a maximum melanin pigment, whereas recessive means less amount of recessive condition will give you an recessive individual with white color and absence of melanin pigment. The dark color is due to the absence of the melanin pigment, whereas white color is due to the presence of the melanin pigment, and in between whatever may be the intermediate will be a 50% which contain the melanin pigment in them. Either they may be a intermediate white. To explain this, you may see here there is a cross with I have written the genes like AABBCC, which is crossed with again AABBCC, whereas one is capital and another one is small letters. The gametes which are produced will be ABC capital and ABC small. When these two are crossed with one another, you can see the F1 generation with capital A, small a, capital B, small b, capital C and small c, indicating the F1 generation to be an intermediate white where the melanin pigment is 50% present. They will show an intermediate color between the white and the black. The other examples to clearly understand the polygenic inheritance are the wheat color in kernel and the eye color which is seen in the human beings. The F2 generation cross is very similar to the other crosses which you have done till now. Please, you can go through the crosses and check whether you will get the correct proportion of an intermediates or the offsprings in the F2 generation. Today we have come to an end of the topics which are related to the certain parts of the genetics. So I'll fastly recap what we have studied in this part. Mainly we have studied about the structure of the DNA, and we have also studied about the structure of the RNA and multiple factors like the gene interaction, where we have studied the multiple factors with an example of a blood group, and even we have solved few problems and have given you certain exercise for you people to solve also. Then we have studied about the polygenic inheritance with an example of the skin color.